Hi, everyone. Thanks for listening in. My name is David James. I'm a graduate student in Juliet Dahlman's lab at the University of Miami. I'm going to be talking about some of my recent work on modeling GI distress and autism. So I think autism spectrum disorder is probably best understood through its diagnostic criteria. Disturbances in communication, restricted or repetitive behaviors, intellectual disability. But in addition to these behavioral issues, there's a whole slew of comorbidities or co-occurring symptoms, including the one that I focus on, gastrointestinal or GI distress. GI distress occurs anywhere from 50 to 75% of the time in individuals with autism and can be really variable in its intensity. Anything from something as mundane as nausea or upset stomach to something as severe as cyclical vomiting, recurring cases of diarrhea or constipation. So I look at GI distress through the lens of two ASD-related genes, SHANK3 and SYNGAP1. I think that using genetically defined forms of autism offers a really good foundation for research that's aimed at answering questions about possible mechanisms for some of these GI phenotypes that we're seeing. So before I get into some of my data, I do want to give a little bit of background information. I think this is probably a figure that everyone's very familiar with. We're certainly all familiar that we have a brain and that it's comprised of many billions of neurons, which collectively have many trillions of synapses. And at these synapses, there's a collection of players that impact form and function. Again, the two that we focus on are SYNGAP1 and SHANK3. These are postsynaptic density proteins involved in anchoring other players in place and making sure that the receiving side of the synapse is functioning the way it should. I think traditionally, and certainly by way of the majority of the research, our understanding of these genes is under the context or within the context of the CNS. But we have this secondary brain, as it's kind of frequently termed, in our gut called the enteric nervous system. And it functions off a lot of these same players and can actually function autonomously without any input from the brain. And then on top of that, recent work has shown that a lot of ASD genes, the expression levels of these genes is actually upregulated in gut epithelial tissue, so non-neural tissue. So Sauer's paper in 2019 shows upregulation of these genes, including SHANK3 and SYNGAP1, within gut epithelium of mice. So there is in all probability roles for some of these genes that are non-neural. So we use zebrafish as our animal model of choice. I think in addition to kind of all the typical benefits that animal models provide researchers, zebrafish have the nice benefit of being transparent for the first couple of weeks of development, which makes studying things like the gastrointestinal tract really easy. So you can see in this magnification in the bottom half of the picture with the adult swimming around up top and the magnified image on the bottom. The gastrointestinal tract doing its thing, going through peristaltic muscular contractions, really rhythmically moving food through its digestive tract. This large black mass is called the swim bladder, helps the fish maintain buoyancy. So I'm broadly interested in the gut-brain axis, how these bidirectional interactions between the gut and the brain might be responsible for some of these GI phenotypes that we're seeing. Specifically today, I'm going to be talking about how mutations in these two genes, SHANK3 and SYNGAP1, might impact GI function. So one of the earlier things that we saw was a change in the rate and the strength of these peristaltic contractions. Peristalsis, again, is rhythmic muscular contractions that moves digestive food matter through the GI tract. So we were able to write a program that was able to record and measure these contractions. And what we found was differences in both SHANK3 and SYNGAP1 mutants as compared to wild-type zebrafish, but for different reasons. So you can see the SHANK3 mutant, the rate and the strength of these peristaltic contractions is reduced as compared to the wild-type. But the SYNGAP fish has a more regular rate of peristalsis, but the strength of the peristalsis itself is actually reduced. So again, SHANK3 is really the only fish showing a change in rate of peristalsis. And the SynGAP fish don't show any significant change as compared to wild type until we look at digestive transit time, or the amount of time it takes these fish to move food through their digestive tract. So in order to get at that, we fed these fish little fluorescent microspheres and tracked how long it took them to pass these spheres through their digestive tract. So on the left, you can see a typical wild type fish. Again, really robust, rhythmic muscular contractions, peristalsis moving these beads through the digestive tract. On the right, you have an example of a shank 3 fish. I unfortunately don't have any videos of a SynGAP fish, but what you see is a drastic reduction in the rate at which these beads are moving through the digestive tract. So in dividing the digestive tract of these fish into 5 distinct regions, dark blue being the pharynx or the throat, orange being the intestinal bulb or the stomach, cyan being upper intestine, yellow being lower intestine, and green being expelled, we were able to track and record how long it took these varying genetic backgrounds to pass these beads. So for these graphs, what you can see is varying colors corresponding to the region of the GI tract. Each color has 3 different columns for the 3 genetic backgrounds, wild type, shank 3, and SynGAP. And then you have 4 different graphs representing the 4 different time points that we looked at: 3 hours, 6 hours, 12, and 24. So you can see in the 3-hour period, all the fish, irrespective of their genetic background, the majority of the beads exist somewhere in between the intestinal bulb and the upper intestine. If you carry this timetable out to 12 hours, you can see that only a few of the wild type fish had any beads remaining in the intestinal bulb or the stomach, whereas the majority of them were actually already expelled. But the 2 mutant models, the shank 3 and the SynGAP fish, were having a much harder time moving the beads from their stomach into their upper and lower intestine and eventually expelling them. So the rate at which these 2 mutants, the shank 3 and SynGAP mutants, were able to pass these beads was drastically reduced as compared to wild type. So we kind of wanted to get at the question of why these mutations might be impacting GI transit time through some potential physiological experiments. And one of the most approachable ones that we had on hand was just to do some dissections and some staining and look at different cell populations and see what might be changing. Our initial hypothesis was, given shank 3 and SynGAP's role in the CNS, this should be a pretty clear-cut case of alterations in the enteric nervous system causing digestive problems. So we stained for HUC, which is a basic neural marker, and then we also stained for 5-HT or serotonin. This was to mark an endodermally derived cell line called enteroendocrine cells. This is a really fascinating subset of cells that I could probably spend my entire time talking about. Suffice it to say, they're secretory cells, sensory cells. They interface with the enteric nervous system. They change their morphology based on what you've eaten, so they tend to pull back when you eat high-fat diets and extend, become a little bit more sharply shaped when you're not eating high-fat diets. So a really interesting subset of cells that plays a lot of interesting regulatory roles within the GI tract. So we wanted to look at that. Contrary to our hypothesis, what we found was that there isn't really a significant difference in the number of enteric neurons within the mutants as compared to the wild type. So if you look at this top portion of the graph, the SynGAP fish are on the right, and then we're comparing wild type to heterozygous and homozygous shank 3 mutants. So slight downward trend in the number of enteric neurons in the shank 3 fish as compared to wild type. Not really that robust a relationship with the SynGAP. Where the significant differences did arise were down here in these enteroendocrine cells. Again, these are endodermally derived cells, so it's really interesting and kind of perplexing as to why mutations in genes that are traditionally understood to play roles in the central nervous system or in the nervous system in general might be impacting endodermal non-neural tissue. And we see a reduction in these cells both in shank 3 fish and SynGAP So this is a shared phenotype that is pretty interesting. So this obviously leaves us with a slew of more questions unanswered than answered, particularly what kind of implications this might have for future research for us, and how the CNS might or might not be involved in these GI phenotypes or GI regulation in general. And we're looking to answer these questions through another cool set of experiments, tissue transplantation. So it's another advantage of using zebrafish is you can take cells from developing embryos from a donor embryo, transplant them into a recipient embryo, and have these distinct cell populations grow up. So there's been a ton of work to date done on getting fate maps for various regions. This figure here is from Catherine Wu's paper with Fraser that maps out brain regions in the developing embryo really well. So we're able to take these cells from one embryo and inject them into another and get these nice chimeric animals. So these animals are otherwise mutant, so either Shankly or Syngap mutant fish, but they have these fluorescently labeled wild-type tissue subsets within various regions of the brain. So with this experiment, we can start to poke at questions about cell autonomy, about how different substructures in the brain might impact other regions of the body. There are a ton of regulatory regions in the hindbrain that play a role in GI regulation. So to be able to go in there and restore the function of either of these two genes, SHANK3 or SYNGAP, really gives us a powerful tool for figuring out what kind of impact it might have on other organ systems. And this obviously leaves the potential for it to not have any impact, which would still be very telling, meaning that these GI phenotypes were arising independently of any issues in the CNS. So briefly to kind of conclude, I want to bring up a few key points. One, mutations in genes traditionally understood to be CNS specific are having impacts on non-CNS tissue. These changes in endodermally derived tissues, these changes in these enteroendocrine cells, point to a possible non-neural role for genes like SHANK3 or SYNGAP1, and could certainly be a source for the GI distress that we're seeing really frequently in autism. Future transplantation experiments will obviously be needed to kind of figure out how or if the CNS is involved in these GI phenotypes. It's something I unfortunately didn't get time to talk about in depth, is the use of these model systems, be it rodent or be it zebrafish, are really helpful in determining what kind of medications can be used to not only treat behavioral issues, but not worsen other problematic comorbidities. A lot of current drugs on the market aimed at treating behavioral issues within the autism spectrum disorder kind of sphere really have negative side effects on GI function. So to be able to use these animal models moving forward and to remember to also check for other problematic comorbidities is really an important part to remember and keep in mind. With that, I'd obviously like to thank everyone in my lab and people who have collaborated and contributed to our work thus far, funding sources included, and I'm happy to take any questions.